For the opportunity to speak today. My name is Daniel Putnam, and I'm excited to have this opportunity to talk about the work we've done at St. Jude Children's Research Hospital to develop an RNA expression-based classifier for pediatric malignancies. Let's see if I can. I want to start, some time ago I was at a conference in San Diego, and I was sitting at a table, and there was a researcher that came up and was very serious and somber and said, "You computational biologists, how are you going to ever you always think your models are correct. How do you know which is right?" And I looked at this person, and George Bach saved my life. I said, "All models are wrong. Some are useful." And then I opened up a discussion with a mathematician by Richard Hamming, where I talked about the purpose of computing is insight, not numbers. And my hope today is I can show the insights that we've gained in building a classifier for pediatric tumor classification. To begin, I want to talk about a patient's experience from symptoms to treatments. Imagine a young child presents at the hospital with lumps, pain, irritability. You go through an assessment process. Following the assessment, that can include molecular and cytogenetic diagnoses, advanced molecular cytogenetic diagnoses, including target RNA sequencing. And ultimately, it would lead to a treatment protocol, which can include chemotherapy, radiation oncology, and/or surgery. And the time from symptoms to diagnosis treatment can be anywhere from 3 to 4 weeks. And the question we are asking ourselves at St. Jude Children's Research Hospital is, can we decrease the time frame between symptoms and treatment protocol? Can we develop a classifier to give insights to say, this patient has a high likelihood to be presenting with this type of cancer? Recently, a paper in 2018 in Nature came out and showed that epigenetic methylation profiles can distinguish tumor types in the central nervous system. And here I'm showing their t-SNE plot on the left, showing that these tumor types group into clusters, and they developed a classifier that could, with high accuracy, classify these tumor types. And we wanted to extend this work to not include just central nervous system solid tumors, but non-central nervous system solid tumors, as well as hematologic malignancies, where fusion status could be really important. In that sense, we used RNAseq expression data. And so I want to talk a little about the sample collection and preparation for our classifier. We collated over 2,000 hematologic and 800 non-central nervous system solid tumor RNA-seq data and combined the RNA-seq data captured from both poly-A mRNA-seq and total RNA-seq data protocols. Just for a quick review, input RNA consists of ribosomal RNA, poly-A mRNA, link RNAs, microRNAs that contain the poly-A tail, and there could be RNA without a poly-A tail. A poly-A filtered method will only include the poly-A tailed RNA sequences, while total filtered will include RNA sequences with and without a poly-A tail. Both methods have filtered out ribosomal RNA. Our method is agnostic to either capture method. We used data from our St. Jude Cloud cohort as an internal data set, and we combined that with external data sets for validation. One was the target data set, and the other was the Australian Zero Childhood Cancer Consortium. And what I'm showing here is we have over 600 harmonized samples from the St. Jude Cloud data and 36 what I'll call not otherwise specified samples. From the target, we had 451, and from the Australian Zero cohort, 48. For our leukemia classifier, we had 3 major classes, AML, BALL, and TAL, and also the NOS category. For our solid tumor classifier, I'm showing the breakdown of the number of samples that we had in each of the class for the St. Jude Cloud data set, the target data set, and the Zero data set. And we see that we have a nice representation of samples from the St. Jude Cloud data set, whereas the target and Zero sets are specific to particular tumor types. Our leukemia class labels are clinically relevant. We have subclasses inside the AML, BALL, and TALL class labels. I want to just mention here that I am emphasizing clinically relevant classes. We have a clinically relevant group that's BALL near haploid and BAL hyperdiploid that have almost identical gene expression profiles in RNA-seq. And so trying to build a classifier to distinguish these two types is nearly impossible. But rather than merge them together, we wanted to show the strengths and weaknesses of a classifier. And so we left the clinically relevant classes in. There are multiple processing steps. We divided samples into a stratified training and test set. We removed a small number of protein coding genes with consistent and large differences between mRNA and total platforms. And then we implemented a normalization method as well as a batch correction method for input into our classifier. So now I want to just talk about our classification method. I'll show some highlights of our pipeline. I'm not going to go through each one of these, but just to briefly summarize this, we take a raw RNA-seq count matrix. The genes are filtered, so we have a filtered count matrix. It goes through our normalization technique, our batch correction method, and then we have, after that's done, we're still dealing with nearly 18,000 genes, protein coding genes. From there, we did reduce the feature matrix, and I'm going to talk more about that on the next slide. And then once we have reduced the feature matrix to a set of features to be fed into our classifier, it's a stacked ensemble classifier consisting of 5 individual base classifiers and then one logistic regression model to weight each of the individual predictions from the 5 base classifiers to give a probabilistic class assignment. So as an output, we get what class does this tumor belong in and what's the confidence score that our algorithm believes it belongs to that particular class. Using our training set from the St. Jude Cloud data, we did bootstrapping, and I created 300 individual bootstrap models and combined them, and I'm summarizing all of this here to identify which feature set we can use. The feature sets we tested was principal component analysis, and the naming scheme is principal component analysis 70 represents the principal components that describe 70% of the variance, and that moved our nearly 18,000 genes to 35 principal components for our solid tumor cohort. And then it goes PCA 75, 80, 85, 90, all the way to 99. And then we also used the top 1,000 most variable genes. And what we see for the solid tumor is there's a cluster of methods that perform well, but as we increase the number of features, we start seeing a decrease in performance, which could indicate we're starting to pick up more noise than features that are relevant to being able to distinguish tumor classes. If we zoom in on the far right, we can see that the top performing method is PCA 75 for solid tumor classifier. For the leukemia classifier, our top performing feature set was PCA 85. Using these feature sets, we then generated that on our overall data set and then built a model. So now we're going to talk about the results that we had. I want to emphasize in our test data sets, I had a test set that had not been seen by any of the training samples from our internal St. Jude Cloud data set, and then we had also 2 external data sets, the target data set and the zero data set. We see that the St. Jude Cloud solid tumor test prediction were accurate. The overall accuracy was 0.994. There was 7 samples that fell below the scoring threshold where we said that the confidence that our classifier had in the prediction was not strong enough, so we just said we're not confident in making a call. And there was a total of 170 samples. And what we see in this graph, and for all of the subsequent graphs in this presentation is going to be as similar, is on the y-axis represents the true class. On the x-axis, we see the predicted class. Along the diagonal is where the true class and the predicted class overlap. Anything on the off-diagonal would be a misclassification. Shown on the right are the number of samples that we have per each class, where orange represents a correct prediction, green represents incorrect prediction, and blue represents samples that fell below the threshold, our scoring threshold. For our target data set, our overall accuracy was 0.987 with 1 filtered sample, total samples 451. And for the, it doesn't want to, okay, all right. For our 0 data set, the overall accuracy was 1. There was 9 samples that were filtered, but for every sample that wasn't filtered, the classifier was correct. Now, moving on to the St. Jude Cloud leukemia test set, it still maintained a high accuracy of 0.925. There were 25 filtered samples with 504 total samples. Here, I will point out, as I mentioned earlier in the talk, that the BALL hyper and BAL haploid, near haploid, had a challenging time in the prediction. I'll just show right here. If we look at the external target leukemia set, we see the overall accuracy at 0.93. There are 78 filter samples with a total samples of 516. So I want to show a case study. This was really exciting for us. If we look at our input data, we see for our rhabdoid tumors, we only had 6 samples from our St. Jude Cloud test cohort, and we had 8 samples from our 0 test cohort. These are not a lot of samples to build a classifier, so we combined our St. Jude Cloud with the 0 to see if we could predict our target samples. In doing that, we can see that for target, we had 65 samples. We predicted 64 of them to be correct, and we missed 1. The 1 sample that we missed is right here, where the true class was rhabdoid tumor, and we predicted it to be neuroblastoma. And we thought, that's interesting. What's going on here? So if we looked at our prediction probability, we see that the prediction probability was very high at 0.975. And so we wanted to look at this by t-SNE, and we see plotted in red are the target rhabdoid tumor groups. In blue are the target neuroblastoma groups, and in yellow is the outlier sample, and it clustered with the neuroblastoma samples. Looking at markers for rhabdoid tumor, we see SMARC-B1 and SMARC-A4 to a degree are downregulated in rhabdoid tumor. And so if we look along the x-axis, we see the target MBL expression violin plot, the neuroblastoma violin plot, and then the rhabdoid tumor cohort in green, and in red is the St. Jude 0 combined rhabdoid tumors. And what we see is SMARC-B1 is upregulated in our outlier sample, and it matches the neuroblastoma group. And then if we look at the MYC-N, which is known, if MYC-N high amplification is a poor prognostic for neuroblastoma, we see that it's highly, highly expressed in our outlier sample, which indicates it could be a neuroblastoma group. Furthermore, in studying the literature, we found a study in 2016 in Cancer Cell where this group, Chun et al., was clustering H3K27 trimethylation promoter densities, and they noted that this particular sample did not cluster with the it didn't cluster with the rhabdoid tumors, but rather the positive controls. And they also mentioned that it expressed both SMARC-B1 and SMARC-A4 being high expressed. And so our classifier identified this sample and said it thinks this sample is neuroblastoma rather than rhabdoid tumor. I'd like to conclude my talk by emphasizing the fact that we built an accurate pan-cancer classification scheme for pediatric malignancies. It really focused on insight and rather than focusing on just the numbers. And this work represents a valuable approach to support tumor diagnosis. With that, I'd like to thank the Department of Computational Biology at St. Jude Children's Research Hospital, particularly Shang Chen and the Department of Pathology. And with that, I will turn it over time for questions.
Thank you, Daniel, for a great talk. So we have 5 minutes for question and answer. So if you have any question, please approach the microphone. So please.
Yeah, thank you so much for the great talk. I have 3 little questions. So first of all is that it seems like, to me, it's more like a multi-class classifier. Could you please just explain the training algorithm that you used in your project?
So the last part, what was the
training algorithm.
The training algorithm?
Yeah.
So you're absolutely correct. It is a multi-class classification problem. And we use a stacked ensemble model where we had 5 individual based classifiers that made a prediction. And then on top of those classifiers, we had another logistic regression model that was trained to figure out the appropriate weights for each of the 5 individual base classifiers. This type of approach is good when each of the base classifiers are a strong predictor if they have strong results individually.
Yeah, so can I understand it's more like 2 layers? The first layer is like 5 classifiers, and the second layer is to choose between them.
It chooses the weights between them.
Weight between them. Okay, thank you. So the second question is that it seems, because I work on blood cancer, it seems that like you have 3 different subtypes of blood cancers. When you train your model, did you treat the data differently, or you just combine them together?
When I trained a model for the solid tumor group and then the leukemia group. But once it was put in a leukemia group, all of the different subtypes within leukemia were treated the same.
Treated the same. Okay, thank you. And the last question is that it seems that you combine different cohorts that together, and you divided them into a 70% training site and 30% test site, right? And have you considered about like use totally independent test set to prove that your result is?
Yeah, that's a great point. So I want to emphasize that for the vast majority of all the samples, we only use the St. Jude Cloud data to divide into 70% training and 30% test. The other data sets were left out untouched, and so they could be a completely independent test set.
Yeah. Thank you so much. Yeah.
Hi, Daniel. Thanks for the great talk. If you go back to the heat map with the target and the 0 data sets, that'd be great. I have a question on how you evaluate performance.
I don't know if I can go back to slides.
Okay, they killed the thing. Okay. Doesn't matter. So comparing the performance between your initial data sets and then when you start talking about target and zeros, we can see that there is, I think they gave you back the AV support today. Great. Yeah, okay. So if we compare this one, for example, the SJ Cloud, but the target is the same with the previous ones, so we can see that there's more fuzziness on this data set, which is fair. But my question is, do you have any insights of why this particular data set is more fuzzy? Is it because there's much more classes, or there are some particular characteristics of the data itself that makes it harder for the classifier to pick up?
I would say a little bit of both. We are dealing with much more classes, but we are dealing with sometimes, I'll give you an example where, as I mentioned, the expression profile for the near haplogroups and the hyperdiploid groups are exactly the same, or nearly the same. And so it's very, very difficult for the classifier to distinguish those groups. Furthermore, if we look at LMO2, LMO2, and TAL1, they have very, very similar expression pathways as well, and it's difficult for our classifier to distinguish that. But we purposefully did not want to merge these groups together. We wanted to keep them distinct because we wanted clinically relevant types rather than just types we knew that we could classify.
Right. I just have a lightning follow-up. When you mention incorrect predictions, it seems to me you're mostly focusing on false or misclassified, but are you more concerned on the false positives, meaning that you predict a certain type of tumor, but it's different, or the false negatives, meaning you say this has no tumor whatsoever? So perhaps another way to put my question is there a negative class when there's like healthy, or that is assumed to belong to any of these classes?
I mean, that's a great question. I think we'd want to have both. We want to capture both of those situations. But I think in this case, we definitely, if we don't want to miss it if somebody has it.
Okay, thanks.
Hi, thanks for a great talk. It's always nice to see good work on classification.
Thank you.
I just had a question about the interpretability of the results, in that, if I seem to recall, you said that the values that were fed into the final model were sort of PCA 45 or something like that. Are you able to, because when you're doing a classification for a medical intervention potentially, it's all the more important that you be able to explain to the patient exactly why you're recommending a particular intervention or a particular diagnosis. So are you able to sort of go back from the PCAs you've got and specify, okay, because I have seen this in the biology, we are recommending this intervention?
I think the way we go back is when we have a strong prediction for a particular class, we can go back to the gene expression and look at what are the canonical, what genes do we expect to be upregulated in a particular tumor type, and we can then say, does that match that? And then that's one way we can look at that.
Okay, thank you.